All right, let's jump right into this. Today, we're going to unpack a really fascinating 2004 paper by Michael Behe and David Snoke. What they did was build a computer simulation to model one of evolution's biggest, most interesting questions. How does nature build new, complex things from basically nothing? And that question right there, that's the absolute heart of the problem. You see, a single mutation that gives an advantage, that's easy to understand. But what happens when a new feature needs 2 or 3 or maybe even 6 different mutations before it even starts to work? How does evolution cross that chasm when all those intermediate steps might not offer any benefit at all? This is the exact puzzle the paper sets out to explore. So to even start answering that, we need to talk about one of evolution's most powerful tools. It's a process that a lot of scientists believe is a main driver of innovation in biology. You can think of it as evolution's copy and paste function, gene duplication. So what exactly is gene duplication? Well, it's pretty much what it sounds like. A gene just gets copied by accident. Now, the brilliant part about this is that the original gene can just keep on doing its important everyday job. And that means the new copy, the duplicate, is basically free from the usual pressures of natural selection. It becomes a kind of genetic sandbox, a rough draft where mutations can pile up without the risk of breaking something the organism needs to survive. And the process itself is so powerful because it's so simple. You've got your original gene doing its thing. Then boom, a redundant copy is made. Now that original keeps the lights on while the spare is totally free to change. This is the raw material, the genetic clay, for new functions to emerge. Okay, but just having this raw material is only the beginning. This is where we circle right back to our main problem. What happens when creating something new isn't just one simple tweak away, but it requires several specific changes to happen, all in the right places? You know, this quote here from the textbook Molecular Evolution just nails the challenge perfectly. If the first few mutations on the path to a new function don't actually do anything useful yet, then natural selection is completely blind to them. There's no advantage to select for, so these mutations are basically invisible, drifting randomly until that very last piece of the puzzle finally clicks into place. The paper actually gives these complex features a specific name, multi-residue features, or MR features for short. And I love the analogy they use. Think of a lock. One pin falling into the right place does nothing. 2 might not be enough. You need all the pins to line up just right before that lock will open. It's the exact same idea for a lot of functions in a protein. And listen, this isn't just some abstract theoretical concept. There are tons of real-world examples. Disulfide bonds, for instance, are like these tiny molecular staples that hold a protein in the right shape, and you need at least 2 specific mutations to create one. Or think about a binding site, that little pocket on a protein where a hormone or a drug has to fit perfectly. That pocket is formed by multiple different points of contact, and they all have to be in the right place for it to work. So to dig into this problem, the authors set up a virtual experiment. And honestly, the best way to think about their simulation is as this high-stakes race against the clock. Imagine a duplicated gene is on this quest to gain a new power, but it's a super dangerous journey. Okay, let's look at this diagram from the paper because it explains the race perfectly. The duplicated gene has to accumulate a bunch of specific compatible mutations. Those are the little plus signs. But at the exact same time, any random mutation pretty much anywhere else could just break the gene completely. That's the big red X. The paper calls this a null mutation. The whole game is to get all the pluses before a single X knocks that gene copy out of the race for. So it all comes down to this really stark contrast, this constant battle. On one hand, you have the goal. Stack up all the necessary compatible mutations in just the right spots to build that new feature. But on the other hand, you have this ever-present danger. Get hit with one random, breaking mutation somewhere else first, and the whole project is toast. The gene becomes useless. Now, to turn this into a real simulation, you have to boil it down to some key variables. So N is just the population size. How many of these duplicated genes are in the running? Lambda, that's the Greek letter lambda, is the number of specific helpful mutations you actually need. And then there's rho, or rho. This one is super critical. It's the ratio of breaking mutations to helpful ones. Basically, it sets the odds of the race. It's the danger level. So the authors set up the race. They defined all the rules, and then they ran the numbers. So what happened? What did their simulation actually find when they applied this model to a realistic scenario in biology? The question really becomes very direct at this point. For a typical protein with a realistic rate of mutation, what kind of population size are we talking about? How much time would it take to actually build one of these multi-residue features? Well, first, they had to estimate the odds. And based on other studies, the authors calculated that for every one specific mutation that might help build a new feature, there are about 1,000 other possible mutations that will just break the protein's function. So the danger of getting hit with that red X is 1,000 times higher than the chance of landing on a helpful plus sign. The odds are not great. And this brings us to one of the most jaw-dropping results in the paper. To evolve a new feature that needs just 6 specific nucleotide changes, the model calculates that you would need a population size of 10 to the power of 22. Let me put that in perspective for you. That number is bigger than the estimated number of stars in the entire observable universe. It's a truly astronomical number. Now, this graph from the paper really shows you why the numbers get so big so fast. Just look at the axes here. As the number of required mutations, lambda, goes up along the bottom, the population size, n, that you need goes up exponentially along the side. It's not a straight line, it's a curve that basically shoots straight up. Every single extra mutation you need makes the problem dramatically, almost unimaginably harder for this specific pathway. So, when you're faced with numbers that big, what do you even do with that? What was the author's conclusion? Does their model suggest that evolving complex features is just some impossibly steep uphill battle? Well, it's actually a little more nuanced than that. What their model is really suggesting is that this one specific pathway they looked at, simple gene duplication followed by a series of random point mutations, is an incredibly difficult road to travel. For most species, especially animals and plants that don't have planet-sized populations, this path alone would require just prohibitive amounts of time to build features that need even a few mutations. So let's just boil it down to the key takeaways here. First, this simple, direct pathway probably isn't the main way things get built in multicellular life, things like us, because we just don't have the numbers. Second, get this. Even a really simple 2-mutation feature would need a population of 1 billion individuals to show up in about 100 million years. So the authors conclude that we have to be looking for other, more complex mechanisms to explain how most of these MR features actually came to be. And this quote is really the punchline, and it comes straight from the authors. They say that we should be looking at more complicated pathways, things like genetic recombination, maybe selection for intermediate steps that have some partial, different function, or other mechanisms we haven't even thought of. So this paper isn't an argument against evolution. It's an argument that this one very simple, very direct pathway is just not enough on its own to explain the complexity we see all around us in biology. Which, of course, leaves us with a really fascinating question to think about. This model shows that the simplest, most straightforward route to building complex things is, ironically, an incredibly difficult one. It really forces us to ask, what other, more clever, maybe more complex evolutionary shortcuts has nature figured out over billions of years to build the incredible machinery of life?